Welcome back to the Explainer. Okay, today we're diving deep, like microscopic deep, inside the fundamental unit of life to look at a system that is absolutely vital for every single eukaryotic organism out there. We are talking about the cytoskeleton. Now, you might hear the word skeleton and think of the rigid, static bones inside our bodies, but the cytoskeleton, it's a breathtakingly dynamic framework. It's the living infrastructure that lets a single-celled amoeba actually crawl around. It perfectly separates DNA when a cell divides, and it quite literally holds your complex human tissues together. Okay, let's dive into this. Just look at this glowing, two-dimensional network. This fluorescent micrograph perfectly captures the sheer complexity of the cell's infrastructure. It is definitely not just some inert, boring scaffold. It's a highly active, constantly remodeling meshwork, spanning the entire cytoplasm, orchestrating this beautiful chaos. It's providing the physical tracks for molecular transport, the structural support to keep the cell from being crushed, and the literal muscle for movement. Here is our architectural blueprint for the day. 1. The dynamic cellular framework. 2. Microtubules. 3. Microfilaments. 4. Intermediate filaments. 5. Cell motility in action. And finally, 6. The evolutionary leap. Let's kick things off with part 1, the dynamic cellular framework. So, right off the bat, completely ditch the idea of a fixed, rigid skeleton. The cellular framework is in a state of constant flux, and it relies on 3 main filament types. Each one has a totally distinct size, core protein, and architectural purpose. At the top, we have microtubules. They're the thickest, sitting at 25 nanometers, acting as the anti-compression highways. Then at the bottom, we've got microfilaments. These are the thinnest at just 7 nanometers, and they act like flexible muscles. And right there in the middle are the intermediate filaments. At 10 to 12 nanometers, think of them as the heavy-duty rebar providing crucial tensile strength. Moving on to part 2, microtubules, the cellular highways. Being the thickest filaments we have, microtubules serve as the cell's vital internal railway system. Picture these as 25 nanometer hollow rigid cylinders. They are built from alternating alpha and beta tubulent proteins that stack up into these little pairs called heterodimers. 13 of these linear protofilaments arrange themselves in a perfect circle to form that hollow tube. Because of this wide tubular construction, they are incredibly rigid. They're the primary anti-compressive struts keeping the cell's shape. But building these tracks requires energy. They strictly depend on binding and breaking down a nucleotide called GTP. So the crucial point is, because of that GTP requirement, microtubules don't just sit there doing nothing. They experience this wild phenomenon called dynamic instability. They are constantly probing the cytoplasm. They'll rapidly grow outwards in a phase called rescue. And then bam, they violently peel apart and shrink in a phase literally called catastrophe. This extreme, rapid-fire behavior is exactly what allows the cell to instantly remodel its railway network, to explore its environment, or grab onto chromosomes during cell division. And what's actually running on these tracks? Highly specialized motor proteins. You see, microtubules have a structural polarity. There's a plus end that grows really fast out toward the edge, and a minus end anchored near the center of the cell. Kinesin motor proteins act like tiny delivery trucks, driving specifically towards that plus end, hauling vesicles and organelles to the outer edges. Conversely, dynein motor proteins walk in the exact opposite direction, bringing materials back toward the center. This incredibly efficient bidirectional highway system is literally what makes large, complex cells possible. All right, part 3, microfilaments, the cell's muscles. Let's shift gears to the thinnest fibers in our tour. Microfilaments are super narrow, measuring just 7 to 9 nanometers thick. Instead of hollow tubes, these are solid, highly flexible cords made from 2 twisted strands of actin polymers. Because they're so narrow, they bend really easily, which is exactly what the cell wants for mobility. They are heavily concentrated right beneath the plasma membrane in a zone called the cell cortex. And just like microtubules needed GTP, microfilaments have their own strict energy requirement. They need ATP to assemble. And that ATP dependency drives a truly fascinating dynamic. Imagine a physical treadmill at the gym. The belt is moving fast, but the machine itself stays in the exact same spot. That is perfectly analogous to what microfilaments do, a process we actually call treadmilling. New actin monomers are added to the front, the plus end, while old monomers simultaneously fall off the back at the minus end. So the overall filament stays the exact same length, but the physical material itself is constantly flowing forward. This continuous forward growth is how the cell physically shoves its outer membrane outward to crawl. And this brilliantly illustrates what that looks like in action. You get this structure called a lamellopodium, this flat, veil-shaped membrane extension. Look at the direction of motion here. That entire leading edge is being physically forced outward by sheer, unadulterated actin power. You have thousands of actin microfilaments treadmilling and polymerizing right against the inner membrane, quite literally pushing the cell forward through its environment. Which brings us to part 4, intermediate filaments, the rebar. Falling right in the middle size-wise at 10 to 12 nanometers, these filaments have a radically different architecture. Think heavy-duty woven ropes. The proteins, like the keratins in your skin or the lamins protecting your DNA, wrap around each other in a staggered, anti-parallel way. And because they're woven together like a rope, they uniquely lack any structural polarity. Both ends are functionally identical. So motor proteins can't use them as directional roads at all. Instead, their one and only job is providing exceptional tensile strength. They resist shear forces so your cells don't literally tear apart under mechanical stress.
But here is the absolute kicker regarding their design. Zero nucleotide requirement. No ATP, no GTP, nothing. Their assembly is driven entirely by spontaneous biochemical interactions. This makes intermediate filaments the most energetically inexpensive, yet mechanically durable elements in the whole cell. They are, for all intents and purposes, the free structural reinforcement that keeps our complex tissues glued together.
All right, part 5, let's look at cell motility in action. You've got to witness this incredible display of biological force. This amoeba proteus is undergoing these dramatic worm-like shape changes. It isn't just passively floating around. It is actively forcing its way forward. The cytoplasm is actively flowing, pushing out those stubby three-dimensional projections known as pseudopodia. And this entire breathtaking transformation, it's being driven entirely by those dynamic actin microfilaments we were just talking about. To really understand what you just saw, we can break this classic amoeboid motility cycle down into 3 very precise steps. Step 1, protrusion. That actin network polymerizes rapidly at the leading edge, literally shoving the cell membrane outward. Step 2, adhesion. The newly extended membrane grabs hold of the surface beneath it, locking in with chemical anchoring points. And step 3, contraction. Using motor proteins like myosin, the internal cytoskeleton violently contracts, dragging the entire bulky cell body forward to catch up with the front. Now, what's really interesting about this visual is what's happening beyond that wide veil. Notice these radially aligned, tightly packed bundles shooting outward. Those are filopodia, active cellular feelers. The skeleton framework naturally forms these highly specialized sensory structures as it moves. The cell is out there literally touching and tasting its microscopic environment, deciding exactly which direction it needs to crawl next based on the terrain.
And finally, part 6, the evolutionary leap.
So how on earth did nature invent this unbelievable machinery? Well, the origins are incredibly ancient. Long before complex eukaryotic cells even existed, our tiny prokaryotic bacterial ancestors were already using simpler versions of these proteins. For example, bacteria use a protein called FTZ for cell division, and it shares a shocking structural similarity to our tubulin. They also use MRB to maintain their shape, which is a direct ancestor of our actin. Over billions of years, these ancient bacterial proteins duplicated and diversified. They evolved into the highly robust interlocking scaffolding that ultimately made large, complex, multicellular life a reality. And this evolutionary leap enabled an absolute explosion of diversity. Because eukaryotic cells mastered the microtubule, they could construct these intricate cilia and flagella. Suddenly, you have single cells covered in thousands of microscopic, beating hairs. It allowed them to swim rapidly, sweep up food, and navigate complex aquatic environments with a level of agility that was previously impossible. You know, the sheer wonder of this microscopic world isn't exactly a new feeling. Way back in 1674, when Anthony van Leeuwenhoek first peered through his primitive, handmade microscope at a single drop of pond water, he saw these exact ciliated protozoa. He famously wrote, quote, "The motion of these animalcules in the water was so swift and various, upwards, downwards, and roundabout that it was wonderful to see." It's just incredible to think that all of our rigorous modern mechanical science traces its roots directly back to that moment of sheer, unadulterated human curiosity. Which leaves us right at the edge of what we know today. The cytoskeleton is a masterpiece of biological engineering, but it is far from fully understood. So I want to leave you with this final provocative thought. If our cells are constantly being physically remodeled by these ancient microscopic motors and highways, a dynamic system continuously responding to the mechanical forces around us, what completely undiscovered biophysical mechanisms might be quietly driving the next stage of human evolution? Think about that as you go about your day. Thanks for joining me on this explainer, and